Alright guys, let's bust out some Module 2 revision. My recommendation is that as you get closer to the day of the exam, you've got to be taking a more holistic view of the content that you're revising. And what I'm trying to do mainly is to show you how I would organize the information, not with the aim of this is the answer of how to organize it, but simply to model how you could do this yourself, and it really doesn't take that long. So let's begin. So we're talking OCR. We're talking OCR Module 2, okay? So if we begin with this, what are the main sections in the OCR Module 2? So the first one is cell structure. I won't be going into all of the details, but hopefully towards the end, if the board were bigger and if my pen's smaller and if my camera more high resolution, then what we would have is we'd be able to put like 5 to 10 points at the end of each of all the branches to get the key points. Okay, so you can do that yourself, but this is mainly an organizational exercise. Okay. Okay, so we got cell structure, but what I'm not gonna start filling all this detail in now. What we're gonna do is look at the whole content of Module 2. So next, after cell structure, we have biological molecules. After that, after biological molecules, we have got enzymes. Okay, after enzymes, we have membranes. And after membranes, we have cells, tissues, organization therein. Okay, so once we've got our first layer, remember, top-down approach. So we've got our first layer, then we start to maybe go to the next layer. So in terms of cell structure, we had two main things. The first part was microscopy, and the other part was the ultrastructure of cells, so organelles, the structure of cells comprising organelles and other structures. So ultrastructure, and again we can fill those out as well. So next, in terms of biological molecules, the main things that we had were, we begin with water, we have carbohydrates, okay, we have lipids, we have nucleic acids, DNA and RNA, we have proteins, and somewhere in there we've also got to find room for the ions. Okay, right. I'm going to quickly run out of space here. I always regret doing these kind of maps because invariably always running out of space in some way. Anyway, right, let's talk about enzymes then. Key things about enzymes. So, first thing about enzymes is their structure. Next key thing about enzymes that comes up a lot is how they work. So the mechanisms of their action. So the lock and key hypothesis, the activation energy, all that stuff. So how they work. Okay. The other common thing about the enzymes is factors affecting enzymes. So knowing how the different factors can affect the rate of an enzyme catalyzed reaction, and also being able to explain how that happens. We also have to give a consideration to the practical aspects of enzymes, because enzyme investigations are a good way to assess understanding of enzyme theory, and so we have the practical considerations as well. It's also a good way to assess whether you understand the key ingredients of what makes a good practical. Okay, so we have the enzymes, factors, and so on. Now let's move on to membranes. So again, with membranes, the first key thing is, again, what is the structure of the membrane? Next is, so after we do structure, we look at, in terms of membranes, we look at the different types of transport across membranes. All right. Next, in relation to cells, so we're working on the second layer now, aren't we? Yeah. So still on the second layer, in terms of cells, we had cell cycle. So we look at the overall cell cycle. Then within the cell cycle, actually, shall we just leave that for later? So we have the cell cycle, and you'll see why I'm avoiding certain topics later. So we got cells, and then we have the organization of cells into tissues, etc. And then we also have specializations. Cell specializations. So this is the entirety of module 2 in a broad sense. Okay, now we go even deeper. We're going to branch off, and remember there's still further detail from what I'm producing here, but each of the ends of these branches theoretically shouldn't be more than about 5 to 10 points. So within microscopy, we first looked at, well, actually, the first part of it was cell theory. So you're going to know cell theory. Then we started looking at the types of microscopes. The types of microscopes. And for each type of microscope, you should know basically how to compare. So how to compare the different types of microscopes, their advantages, their disadvantages. All right. So in terms of the comparisons, what were the main things we used to compare? So we're comparing the type. We're comparing types of images. So can you describe the different types of images that they can all produce? We're comparing resolution. We're comparing magnification. Yeah. We're comparing even convenience or practicality. You could replace that with practicality. All right. So we're comparing them, and it's best to try and keep the categories of comparison consistent, so you can easily make the points. Right. Once we look at types, then we kind of looked more in particular at light microscopy, because we need to be aware of the different types of slide preparation. So what are the principles of how we prepare those slides? Okay, so we looked at light microscope, in particular at slide preparation, and building on from that, the usefulness of differential stains. So, okay, so this is going to form the end of my branch here. Is it because I don't know all of that off by heart? Yes. But also, you know, the examples of the differential stains, so maybe I'll just put that on there. So have some examples of the differential stains, know the advantages. So why do we use differential stains? Have some examples of the differential stains and what they bind to. Okay, so the applications. Right. Okay. Then we had, what do we have? Oh, the idea of magnification. So how do we calculate magnification? And within the idea of calculating magnification, also the further idea that we have to have calibration. So knowing how to use the eyepiece graticule, okay? What we're saying is we're going to have to talk a little bit about magnification, or at least be able to use it, okay? But yeah, how the eyepiece graticule is calibrated, etc. I hope we're okay with this so far. Okay, we've kind of gone to the end with microscopy there, and now we'll look at ultrastructure. Now, ultrastructure really is quite straightforward, because in this category all you really need to know is you've got to know all the organelles, but then the more tricky thing is, for each organelle you have to know, you need to be able to describe their structure, you need to be able to recognize them if you see them, and for each of the structures, or for each organelle, you need to know its structure and what its function is. And I'm going to add another arrow here, how the structure might even relate to function. Okay, so for example, if we're talking about the smooth endoplasmic reticulum, we know that its function is to synthesize lipids, for example, or carry out lipid metabolism. But what is the structure part of the smooth endoplasmic reticulum that actually relates to that function? Because no function is possible without the structure. All right? And so there we just need to be able to say that, well, you know, it's got a large surface area of all the membranous or the membrane sacs that make that, or the interconnected membrane sacs that make up the smooth endoplasmic reticulum, makes a high surface area, but also that the smooth endoplasmic reticulum contains the enzymes required to carry out the lipid metabolism. Okay, so that's how structure relates to function. So you've got to be able to state those two for each of the organelles, but also connect those two up together. Moving on. Now the ions, in terms of the ions, not only do I not have a lot of space, but you just got to know the examples, okay? So unfortunately, that's just something you're going to have to learn. Let's move on then with water. Now with water, again, as with a lot of biology, what's its structure? How does the structure connect to the properties of water, and how are the properties of water connected to or important for life? Those three things are very important to not only know individually, so there's, you know, you could write a few points about the structure of water, you could list off its properties, but you also need to know, like, what is it about the structure of water that makes it cohesive? Okay, and then why is cohesiveness important for life? Okay, so in terms of water, know its structure, how its structure results in its properties, i.e. it's a polar molecule, etc. But then why those properties are important for living organisms, and I think that should cover water. In terms of carbohydrates, okay, we've got a number of different types, all right, but primarily, you know, in terms of generally speaking, the monomers that make up the carbohydrates are mostly, from what we look at, is glucose and the different types. So we need to be aware of how alpha and beta structures differ from each other. But once we've sorted that out, then we go on to at least the main points, guys. Please appreciate, you know, I know there's details beyond this, but what we're trying to do is kind of give a rough picture. And you can fill in all the extra details on top afterwards, okay? But I really do think that as we get closer to the exam, we do need to focus more and more on the main ideas rather than the details. So in terms of carbohydrates, there's a few examples that we need to be aware of in detail. So we've looked at our monomer here. Okay, the idea that condensation reactions bring them together, but, okay, so actually, let's just follow that through then. The idea that alpha and beta glucose monomers can be joined together using through condensation reactions forming glycosidic bonds. Okay, then we look at our examples, and again we need to start relating structure to function here. All right, so how shall we do this? Yeah, let's do the examples first. So with carbohydrates, actually there's two types that we look at. So the first type is the energy storage carbohydrates. All right, so the energy storage carbohydrates. So which ones are they? So we have starch in plants, and we have glycogen in animals. Okay, so we see how we're compartmentalizing the information as we go along, and maybe I might have used a different organizational structure, but I'm able to do that because, you know, I know the content, and either way would have been right. It's just however you want to do it, organize it. Okay, so at least there's a pathway to connect and get to certain information when you need it. There's a logic behind it. All right, so there's starch and glycogen. So again, glycogen, key points is that it is alpha glucose, it is highly branched, okay? It does have 1 to 4, 1 to 4, 1 to 6 glycosidic bonds, highly branched. All that structural detail, actually. Okay, again, we do the same thing. So yes, we have, and I might not detail it here, but we have structural detail. But then how does that relate to its function of being energy storage? Okay, so again, note down its structural details, great. Know that its function is energy storage, good. But what's brilliant is if you can say how. How is it that the structure relates to the function of glycogen? Okay, the fact that it's a polysaccharide is insoluble, doesn't affect water potential, tick. The fact that it's highly branched, which makes lots of branching, lots of endpoints for enzymes, or a greater surface area for enzymes to hydrolyze, releasing glucose molecules at a higher rate. Tick. Okay, so that's what we're doing there. Again, with starch, yeah, what are the key structural points with the starch? Okay, amylose, amylopectin, and so on. Okay, and again, with starch, it is also a storage molecule, and so what is it about the structure of starch that makes it a good storage molecule? Okay. Next, we have a structural carbohydrate, which is cellulose. Okay, and so, right, what is it? So what's the key structural points about cellulose? Beta glucose means straight chains joined together with beta 1 to 4 glycosidic bonds, making straight chains which, because they're straight, multiple chains molecules can come together close enough to hydrogen bond, can make bundles, therefore strong. Okay, so you see what we're doing here. So again, we've got structural information. We know that its function is different. Right, how does its structure relate to function? Okay, right, next, lipids. All right, lipids. Actually, I'm going to have to move some things around here. Okay, so let's talk about lipids next. I knew this was going to happen. I just knew it. Right, so we've got lipids. Again, with lipids, I would begin with the triglyceride. So they've got an overall structure, which is the triglyceride. Okay. Then what I would do is then look at how the structure of a triglyceride can be different from, actually no. So we've got triglycerides, okay, and we've got, we appreciate that the triglyceride is made of glycerol and it's made of fatty acids. But we need to appreciate that fatty acids can be saturated or they can be unsaturated, and that makes a difference to their properties. Once we've looked at that, then we can look at phospholipids as a kind of modification of the triglyceride structure. So we just see like what's the key difference in structure between a phospholipid and a triglyceride and how that affects its properties. Okay, so again, triglyceride has certain properties, okay, and those properties result in what functions triglycerides have. Okay, so they are energy storage, they are insulating molecules. What are the properties that allow the triglyceride to have that particular function? Okay, so now we look at phospholipids then. All right, what unique properties does the phospholipid molecule have that a triglyceride doesn't, because the phospholipid is different in structure? Okay, and then we connect that back to, right, so the phospholipid has different properties. What important function can phospholipids have, okay, that triglycerides don't, because they have different properties? Okay, so that's lipids, but I think we are not done with lipids until we also talk about cholesterol. Okay, so the cholesterol, again, key points about its structure. It's difficult to memorize its structure, but just be able to describe its structure, okay, and which unique properties does it give such molecules, and because of those unique properties, well, what particular function can it play in the body because it has those properties? Okay, might link back to some of this stuff when we look at membranes. All right, let us move on. I think that there's one thing that I've been missing here in relation to all this stuff is testing. So with carbohydrates, we need to be able to know which biochemical test to do, to test for example, testing for reducing sugars and testing for starch. Okay, and with the lipids, again, yes, we need to test also with the lipids. Oops, there we go, right there, lipid test. So the emulsion test for lipids need to include like a short section on the key steps we do to carry that out, no more than 4 to 5 points, knowing what the positive test would be, etc. Okay, so that's lipids. And then we move on to proteins, and I'm supposed to fit all of proteins into that space. Not going to happen. So I'm just going to move this around a little bit. Proteins, we've got our monomers. This is more like a line. With proteins, we begin talking about amino acids, what their general structure is, okay, and properties, but then how the amino acids undergo condensation, producing for us, you know, the polypeptide, okay, and that is essentially, or at least understanding that how the polypeptide is related to the primary structure. Okay, so be able to define what the primary structure is, but understand how you got there from your starting material of amino acids. Next, how the primary structure then undergoes, due to hydrogen bonding, okay, how that is connected to secondary structure. Okay, your alpha helix and your beta pleated sheet. So that's the secondary structure. And then how the secondary structure then gives you the tertiary structure because of the involvement of the R groups, okay, and the four types of interaction between the R groups. So the 4 types of interaction between the R groups. Okay, and that gives you the tertiary structure. So let's go from secondary structure to R groups to the 4 types of interactions, 4 types of interactions to the tertiary structure. Okay, and then the appreciation that it doesn't simply end there, because different polypeptides, so multiple polypeptides, so from the tertiary structure we can then go on to the quaternary structure. The appreciation that, again, because of the same kind of R group interactions, those R group interactions can not only hold bits of the same polypeptide chain together, but bits of different polypeptide chains together, therefore giving us different types of quaternary structures. Okay, right, now with the proteins, once we've looked at the overall structure, so let's look at structure here. So you've got the structure. Right, now different types of proteins might also have, as part of their structure, might also be globular, and they might also be fibrous. Right, now globular and fibrous proteins all have different, each have different specific functions. Now globular proteins, you know, what are their properties? So if you're a globular protein, you know, there's a certain common features that you have. What are they? But also globular proteins have specific functions that only globular proteins can carry out. Now globular proteins can have a wide range of functions which we need to be able to state. Fibrous proteins, on the other hand, you know, they have some unique properties as well in terms of structure. But then fibrous proteins are usually only have certain specific functions, usually to do with structure. So structural and supporting function. There may be some exceptions, but there we are. All right, again, I've got myself into a situation where I don't have a lot of space. Right, let's keep moving then. Maybe I'll move the membrane stuff a little bit, but right, in relation to enzymes then, there's a few key things. Structure, that's easy. The key thing about the structure of an enzyme is that it is a protein. So all of this stuff is true, at least usually enzymes are, or enzymes will be globular proteins. So be aware of that. So all of this stuff is true, but then enzymes are globular. Okay? But then also there's a unique thing that they do have, which is the AS, or the active site. Okay? So enzymes have the active site, which is a unique structural property to them. Be aware of that, how it has to complement the substrate, etc. All right, so enzymes structure, it is a protein, it's got the active site. Right, next, how they work. How they work. Now, in terms of how they work, there's a few key things. I always like that we begin with a description or discussion of the activation energy. The discussion of how the enzymes work must begin there. But then there's other things that we can mention. So there's the mechanism, right? And the mechanism can involve lock and key, and it can involve induced fit. Okay? But the other thing that we can't forget about the mechanism is also about the idea that enzymes work because of collisions with their substrate, and how the collisions form the enzyme substrate complex. It's a key part of a lot of the stuff that we do with enzymes, is understanding how all these different factors that might affect enzymes affect collisions, effective collisions that form enzyme substrate complexes. Okay, then we can also move on from here to factors. which factors affect how enzymes work. Oh, again, I've forgotten something. Proteins, how can we test? Okay, how can we test for them? Remember the Biuret test for proteins. And also, remember, we can test for amino acids using chromatography. Okay, so where were we? We're looking at factors that can affect enzymes. So know the factors and be able to explain how they affect the rate of an enzyme controlled reaction, using your idea of the shape of the protein, the shape of the active site, collisions, activation energy, and so on. Okay, so what the factors are: pH, temperature, we have pH, temperature, we can have the concentrations, we can have cofactors that help the enzyme to carry out its function, but we might also have inhibitors that might reduce the activity of the enzyme. And remember, we've got different types of inhibitors as well. But we can also investigate, practical. We can also investigate the effect of these different factors on the enzyme. Remember, with all the practicals, we have to have the independent variable, which will be the factor that you're investigating. We need to have the dependent variable, which will be how you are measuring your reaction. So either you might be able to measure the disappearance of your substrate, you might be able to measure the appearance of your product, or some other change that's happening as a result of the enzyme activity. Finally, you will need control variables. So you'll need to control the other factors that could potentially affect the experiment in some way. Okay? And by keeping those the same, you can then be able to validly investigate the effect of that particular variable. Okay, so we've got those practical considerations. As well as that, remember, once you've got your dependent variable, you've got your results, you'll need to plot that on a graph accurately, all the things that go with that. But then you also need to be able to calculate the rate of the reaction from, or it might be the initial rate. Okay, remember the tangent to the curve at time 0, slope of the line, using the axis units, etc. And I think there we have enzymes. All right, now we move on to membranes. Now with membranes, I think it's a little bit more straightforward. We kind of look at the structure of the membranes, beginning with the phospholipid. Then we look at biological membranes. So we look at phospholipid, but then we also look at why bilayers form. But then the other important part about membranes is the idea that membranes have proteins embedded in them as well, and what the functions of those proteins could be. So we look at biological membranes from that perspective. Then we look at the function. Okay, so in my view, when we look at the function of membranes, we are looking at all the different types of transport that could occur across the membranes. Okay, so we need to be able to discuss simple diffusion, knowing the properties of the membranes and how they are involved in interacting with the properties of the molecules that might be moving across. Okay, and how that has to be done, or how that occurs through different mechanisms depending on how the membrane and the particle or substance interact. Okay, so knowing that non-polar molecules that might be small move by simple diffusion, knowing that some larger molecules that might be polar or charged needs to move by facilitated diffusion with the involvement of proteins. We might even have osmosis occurring through protein channels. We might have active transport, pumping carried out by proteins using the energy available in the cell. And we're going to have the bulk transport exocytosis, endocytosis, and so on. All right, and is that it? I think that's it. Yeah, I knew this was going to happen. I knew it, but I tried it anyway. I forgot about DNA. Right, so DNA, we're going to have to find some space for DNA. Right, okay, we'll continue on from biological molecules. This is so embarrassing. Biological molecules, okay, we'll find some space for DNA right here, or nucleic acids. Okay, so nucleic acids, their basic unit is the nucleotide. Okay, so what's the structure of a nucleotide? The ribose sugar, or the pentose sugar, the nitrogenous base, and the phosphate, and how they're arranged. Nucleotides then undergo condensation to form polynucleotides. Many nucleotides join together through condensation reaction, phosphodiester bond formation. Okay, but then polynucleotides could be of different types. So you could get DNA, right? Now DNA has the base pairing, okay? So the base pairing being C and G, I think, with the 3 bonds, and A and T with the 2 bonds, whereas the polynucleotide in RNA is single-stranded, so base pairing doesn't really apply there, but then the difference in structure that RNA has, so it being single-stranded, it having uracil instead of thymine, etc. Okay, so that's all kind of structural details, but then we also need to state, kind of describe that RNA has a different function. RNA has different functions in terms of forming ribosomes, tRNA, what's the function of the tRNA are in protein synthesis. Okay, so there is that. DNA, structurally, that is DNA, but then what are the functions of DNA? Okay, so I guess when we talk about the functions, shall we just come down here? The functions of DNA and RNA do combine in the process of protein synthesis. So DNA has got 2 functions. One is DNA replication. So we discussed the process of DNA replication. Its other process is protein synthesis, encoding within genes sequence of bases that are the instructions to join amino acids in a particular order. And it's in the protein synthesis part of it that we really get the involvement functionally of RNA as well. So in protein synthesis, we have the functions of RNA, being messenger RNA, being transfer RNA, being in the ribosomes that carry out translation. Okay, so we need to discuss those protein synthetic processes as well. So we have transcription, and we also have translation, and we need to be able to describe those processes. Okay, and then we have DNA replication, i.e. the process that it is involved, or the sequence of events that results in DNA replication. So that is DNA. We've done membranes, and the last bit is then cells. Now in the cells we have the cell cycle, okay, so that's its own thing, but then as part of the cell cycle we need to talk about mitosis as the cell division process, but we also need to talk about meiosis. So cell cycle generally, then the 2 types of cell division. And as a result of mitosis and meiosis and differentiation, you get the different types of cells with different types of specialization. So I'm just going to go from mitosis to specializations. So mitosis and differentiation, so I'll just put that here, because mitosis on its own wouldn't produce new cells, but differentiation does, and that also implies that there are cells that are not specialized yet, i.e. they are stem cells. Okay, but stem cells which undergo differentiation produce specialized cells, and specialized cells, remember, specialized cells and tissues have particular adaptations, particular structures that allow them to perform unique functions. Okay, so we got specialization of structure and we got specialization of function, and we need to relate that back. So for example, the cells of the alveoli are very flat and thin, which reduces the distance of diffusion, i.e. increasing the rate of diffusion for gas exchange in the lungs. That's the kind of thing that we are talking about. And are there other examples? So we need to relate those specializations to their functions. But then, once you do have that, you have tissues that form organs, organs that form organ systems, and organ systems that form fully functioning organisms. All right? And that is that. I think I will stop there. This, again, just like biology, like I hope you're not looking at this and thinking, oh shit, okay, because you know what biology is all about. There is a lot of stuff, but you've had a significant amount of time. You are now, you're ready to organize this stuff. So, you spent a lot of time learning that content. Your next stage before the exam hits is, can you organize it? Because if you can organize it, you know how it's all connected up. You know how you can compartmentalize it, how you can organize it so that at a moment's requirement, you can then get to the bit of information that you need. Okay, and that's what it's all going to be about in a few days' time. All right, guys? So if you don't agree with my structure, the point being, try and do this for yourself. Okay, this is not about what I have created. This is about me showing you how it can be done, you guys doing it, and I hope by you doing it, if I don't get around to doing this for module 3, 4, 5, 6, that's not the point here. The point is, this was how it was done. You can do it too. Do it quickly, and I hope that it's going to make a big difference for your exam. Okay? Good luck, guys. Go and be awesome.